Hello everyone, this is Dr. P. Mary Anupama, Head of the Department of Biochemistry, St. Joseph's College for Women. Today, let us study about electron transport chain. Lesson outcomes. By the end of the course, the learners will be able to understand the localization of each of the component of electron transport chain. Then they'll be able to illustrate the composition and working of each of the components of electron transport chain, explain about the various carriers of electron, then narrate the functions of all the four complexes of electron transport chain. Apart from this, the learners will be able to differentiate between the functioning of each of the complexes, right from complex 1 to complex 4. The contents of this video will include introduction to what is electron transport chain, the components of electron transport chain abbreviated as ETC, then mitochondrial redox carriers, then followed by electron carrier complexes. Let us look into what is electron transport chain. Electron transport chain, it's also called as electron transport system. It comprises of a chain of reactions that convert the redox energy available from the oxidation of NADH and FADH2 into proton motive force, which is used for the synthesis of ATP through the conformational changes in ATP synthase complex through a process which we call it as oxidative phosphorylation. So you see that this particular concept, it starts with oxidation of the compounds macromolecules resulting in the reduction of NAD plus and FAD that leads to NADH plus H plus and FADH2. These actually enter the electron transport chain, and during this process a proton gradient is generated across the inner mitochondrial membrane. That is the driving force for the synthesis of ATP from ADP and pi. Most of the eukaryotic mitochondria, they are involved in this oxidative phosphorylation and then electron transport chain. So you see that all the reactions of oxygen with the products of the citric acid cycle or even the amino acid metabolism or fatty acid metabolism, okay, if they finally end up in conversion to carbon dioxide and water, but during this process the NADH plus H plus formation and as well as FADH2 formation will be taking place. So these enter the electron transport chain, which is located in the inner mitochondrial membrane. So you have a series of components which are present in the inner mitochondrial membrane. So as these electrons are getting transferred across the membrane, finally they end up reducing oxygen to water. So all these are the components that are located, and you can see that some are getting reduced while some are getting oxidized, and finally a proton gradient is generated, which is mainly the sole source for ATP synthesis. Let us see what are these mitochondrial redox carriers. So a total of 5 membrane proteins were identified, and these are involved in the generation of that proton gradient. Of all these 5, 2 they are considered to be mobile electron carriers, while you have 4 complexes also that are involved. So the 2 mobile carriers for the electrons, they are the ubiquinone, abbreviated as Q, and then cytochrome C, abbreviated as CYTC. So during the study we will be using these two terms. So ubiquinone, it is present in the inner mitochondrial membrane of the mitochondria, while cytochrome C, it is attached to the outer surface of the inner mitochondrial membrane. That is how these two are located. Now let us look into each of the components of electron transport chain. So you have first one is nicotinamide nucleotide. So we know that NAD plus is a coenzyme that is involved in electron transport chain, and when it is reduced it is getting converted to NADH plus H plus by a dehydrogenase. These dehydrogenases, they are involved in the removal of two hydrogen atoms from the substrate, and then they are attached to this NAD+. So here the substrates that contribute to NADH plus H plus formation, they are like the glyceraldehyde 3-phosphate or the pyruvate or isocitrate or alpha ketoglutarate and even malate. So all these will be contributing to the formation of NADH plus H plus. Now the second component is flavoproteins. So we have seen NADH dehydrogenase, it's an enzyme, and a flavoprotein with FMN as the prosthetic group. So this itself has FMN as its for prosthetic group. So here FMN, it becomes FMNH2, okay, upon reduction. So here what happens is it accepts two protons and becomes FMNH2. You also have FAD. FAD, it's a coenzyme and it is found associated with succinate dehydrogenase, and it is found in the inner mitochondrial membrane. So what does FAD do is it accepts two hydrogen atoms from succinate. Okay. We also have iron sulfur proteins abbreviated as FES proteins. So there are around 6 iron sulfur proteins associated with the electron transport chain. And these are involved in electron transfer from FMN to coenzyme A and to cytochrome B to cytochrome C1 and so on. Coming to coenzyme Q. Coenzyme Q, it's also called as ubiquinone. It is derived from quinone and it has isoprenoid side chain. And this is a lipophilic electron carrier and it accepts electrons from FMNH2, which is produced from NADH dehydrogenase, and it is studied which is studied previously, or it can also accept electrons from FADH2, which is produced out of electron transport chain. Okay, FMNH2 is a part of this, while FADH2 is produced out of the electron transport chain. So coenzyme Q can accept electrons from anywhere. The last components are the cytochromes. So we know that cytochromes, these are conjugated proteins. They have a heme, and we know that heme is a porphyrin ring that has iron atom at its center. So the iron in this heme of the cytochromes, it exists in oxidized form as well as reduced form. Oxidized form means you have this Fe3 plus and reduced form means Fe2 plus. So this is important for the electron transport chain to operate. That means for the transport of electrons in the electron transport chain, this oxidation and reduction of the cytochrome should be taking place. So that the transfer of electrons from coenzyme Q to cytochromes, it takes place in a path. Like many cytochromes were discovered, initially it was A, B, C, then some of them were discovered, but the flow in which it takes place is first is B, then comes C1, then comes C, followed by A and A3. So this is how the electron flow takes place from coenzyme Q. Now let us study about the most important component of the electron transport chain, that is the electron carrier complexes. So we have 4 electron carrier complexes. First is the complex 1, which you call it as NADH ubiquinone oxidoreductase. Next is complex 2, which we call it as succinate dehydrogenase. Then the complex 3, which you call it as cytochrome BC1 complex, and complex 4, which is cytochrome C oxidase. You also have a complex 5, which is ATP synthase, but that is oxidative phosphorylation. So I have included only 4 main complexes. Once again, complex 1, which is NADH ubiquinone oxidoreductase, complex 2, which is succinate dehydrogenase, complex 3, which is cytochrome BC1 complex, and lastly complex 4, which is cytochrome C oxidase. Let us see each of these one in detail. So before going into details, the simplest representation which I would like to share, NADH plus H plus. So from here it goes to complex 1, then to ubiquinone, followed by complex 3 to cytochrome C, then to complex 4, and then water formation takes place. Here you have complex 2. From succinate FeDH2 directly through the complex 2, it transfers the electrons to ubiquinone and then the flow continues. Complex 1, we also call it as NADH ubiquinone oxidoreductase or type 1 NADH dehydrogenase. So here what you see is that when NADH plus H plus is formed, and as it enters this complex, 2 electrons are removed from this one and they are transferred to the mobile carrier for electrons which is present in the inner mitochondrial membrane, which we call it as ubiquinone, abbreviated as Q. Now, when the electrons are transferred, the ubiquinone gets reduced to ubiquinol QH2. So this mobile carrier, it easily diffuses across the membrane, whereby it will be transferring the electrons from complex 1, okay, and then it goes to the next one. But during this process, what it does is, it will be translocating 4 protons also across the membrane, thereby contributing to proton gradient. So as it is transporting electrons from complex 1 to complex 3, it will be generating proton gradient because it will be pumping 4 protons across the membrane, contributing to proton gradient. So as such, NADH, it is getting oxidized back to NAD plus because the electrons are transferred by the FMNH2 and the 2 electron transfer will be taking place. The FMNH2 is oxidized by transferring electrons to Fe iron sulfur carriers, which will be present here, and then to ubiquinone. So during this entire process, what you see is that 4 electron transfer is taking place into the intermembrane space, or you see that 4 protons are being pumped. So every 2 electron transfer, that would result in the pumping of 4 protons into the inner membrane space or the intermembrane space. So from the inner mitochondrial membrane across the membrane from the matrix, they will be pumped at the complex 1. Next is the complex 2, which is also called as succinate dehydrogenase or succinate coenzyme Q reductase. So succinate, it enters through this complex. This contributes to addition of few more electrons to the ubiquinone or the quinone pool from the succinate. But you see that this pathway, it contributes less of electrons or less of energy to the electron transport chain as compared to NAD+. So here what you see is that when the succinate enters here, it will be transferring electrons to ubiquinone over here. So there is no entry through complex 1. Directly it is going to complex 2, then to ubiquinone, and then to complex 3. Next we have complex 3, which is called as cytochrome BC1 complex. 2 electrons, they are again removed from this QH2 which is formed here, and these are transferred through this mobile carrier to the complex 3. So as a result of this transfer, the QH2 is converted back to Q and returns back to its position. Here it is Q. Once it becomes QH2, it transfers the protons to this complex 3, that is cytochrome BC1 complex. So here, once the electrons are transferred, the 2 protons will be pumped out, leaving an oxidized form of Q, which returns back to the complex 1 for the next cycle. So this is how ubiquinone will be moving QH2 back to Q. And this is the complex 3, wherein you have 2 protons transferred across the membrane. Next is our complex 4, which we call it as cytochrome C oxidase. So 4 electrons are removed from the 4 molecules of cytochrome C and then transferred to the molecular oxygen and 4 protons, thereby resulting in 2 molecules of water formation. That means for each atom of oxygen, 2 hydrogens are consumed, and you see that 2 molecules of water will be formed. Here also, at the cytochrome oxidase complex, protons are pumped into the inner mitochondrial membrane. So these are the 3 sites: complex 1, complex 3, and then complex 4. While when it comes to succinate and FeDH2, it goes like this. Let me summarize the electron transport chain and the complexes. This is the complex 1, which is NADH dehydrogenase, where the NADH plus H plus, which is formed from the oxidation of biomolecules, it enters here, thereby transferring electrons, and the protons are pumped into the inner membrane space or intermembrane space. The protons are taken up by ubiquinone, which is the mobile electron carrier, and this one, it transfers it to the complex 3. Now the complex 3 is cytochrome BC1 complex, wherein again the electrons are transferred to cytochrome, which is an electron carrier, which transfers the electrons to the complex 4. Here also, during this process, protons are pumped. While in the last one, which is the cytochrome oxidase complex or the complex 4, which receives electrons from the cytochromes, here you see that oxygen becomes the acceptor for the protons, and finally it is converted to water. During this process also, as the electrons are transferred, protons are pumped into the inner or the intermembrane space. As the electrons are transferred through the electron transport chain of the inner membrane of mitochondria, you see that protons are accumulated. They are pumped into the intermembrane space from the matrix. So here they are accumulated, and these cannot flow back into the matrix. They require the help of ATP synthesis to do so. So ATP synthesis, what they do is, when they are pumping the protons, here phosphorylation of ADP will be taking place, resulting in the synthesis of ATP. So here ATP synthesis, they are catalyzing the phosphorylation of ADP, and it is driven by the movement of protons across the membrane. So from the intermembrane space, they will be pumped into the matrix, restoring it back to normality. So this is how the ATP synthesis will be taking place after electron transport chain is completed. The references which are used during the preparation of this topic are Leninger's Principles of Biochemistry, then Biochemistry by U. Satyanarayana, and also Harper's Illustrative Biochemistry. Thank you for watching. So we have seen thoroughly what is electron transport chain, the carriers of electron transport chain, the complexes of electron transport chain, and at the end, towards the end, you see that it is the proton gradient which is established, that is driving the synthesis of ATP.